Hello children, welcome for today's biology practical class. So today we are going to study the practical how to separate the plant pigments through paper chromatography. So for this purpose I have taken some freshly plucked leaves, that is Centella asiatica, what you call Vallarai in Tamil. You can take spinach leaves also, mortar and pestle. So here I have taken the ether and acetone in 9:1 ratio as a solvent, and some sand for crushing, capillary tube, split cork, chromatography paper, test tube, and finally the chemicals, ether and the acetone. So now let us start the practical. Now I have taken the leaves, and the leaves are to be crushed with the help of mortar and pestle. So for that purpose, to facilitate better crushing, I am adding some soil. Okay, this is sand. So it facilitates, as it is having rough surface, it will facilitate the crushing properly. So here we can put some acetone. Acetone is a solvent, it dissolves all the pigments. Okay, so crush it properly. After crushing, I have got this much extract, leaf extract. This one I am going to use to do the paper chromatography. A thin strip of paper I have taken. So now with the help of a capillary tube, just I am putting the extract upon this. We have to allow it to dry. See here, this drop, when it is dried, then the other drop you can put. Okay, but you should not dry this in sunlight. It should dry up. Then later you can put the other drop. Like that. You can repeat for 4 to 5 times. Now it can be kept inside the test tube. For that purpose, I have taken 9:1 ratio ether and the acetone. So now I am putting the strip inside. While putting the strip inside, it should not touch the sides, this chromatography paper. Only the tip, it has to be immersed inside the solution. Okay, by means of split cork, we can secure this properly. Okay, so while keeping inside, just you see the drop what you put, that leaf extract, it should not touch the solvent. Only the tip of the chromatography paper it has to touch. Can you see the drop? So here the chromatography paper, it should not touch the sides, only the tip of the chromatography paper only touching the solvent. Now we have to allow this to remain for at least 1 hour. Then only we can see the pigments moving up. The test tube, it has to be closed tightly. It has to be airtight. Otherwise, the solvent will get evaporated. Can you see, children, the movement of the solvent? When the solvent is rising slowly, that green pigments, that will also, that is the chlorophyll and other pigments, they will start to move. We can observe it after few minutes. Children, can you observe the separation of pigments here? If you are doing in spinach leaves, the separation of pigments will be better. Thank you.